students. In last video we have seen various infectious diseases, their symptoms. Now in this video we will start with prevention and control of infectious diseases. Personal hygiene. Keeping body clean, consuming clean drinking water, food, vegetables and fruits is important. Secondly, public hygiene. Proper disposal of waste and excreta is required. Disinfection of water reservoirs should be done. Third point is elimination of vectors and their breeding places. Avoid stagnation of water. As you know, mosquitoes, they grow in stagnant water. Clean household coolers regularly. Use mosquito nets and introduce Gambusia fish in ponds which feed on mosquito larvae. The fourth point is avoid contact with infected persons or their belongings. Use of vaccines and immunization programs. Different vaccines which are available for diphtheria, polio, tetanus. So one can prevent from such diseases by use of vaccines. We will see what is immunity. Immunity is the ability of the host to fight the pathogen conferred by the immune system is called immunity. So our immune system gives us ability when certain foreign particle or pathogen enters in the body to fight against that pathogen and destroy it. Such power is called as immunity. There are two types of immunity: innate immunity and acquired immunity. Innate immunity, it is non-specific type of defense. It means this immunity will work for any type of pathogen, not only for one particular but any type of pathogen, may it be bacteria, virus or any bacteria. That is why it is called as non-specific type of defense. It is present at the time of birth. You get it by birth and is accomplished by different types of barriers to entry of pathogen into our body. There are 4 different types of barriers. With these barriers functioning in our body, we are having this innate immunity which avoid the entry of bacteria or pathogens in our body. Now, the 4 types of barriers are: physical barrier. Skin is the main barrier. It prevents entry of microorganisms into the body. The another one is mucous coating of epithelium lining of respiratory, gastrointestinal and urinogenital tracts. These mucous linings help in trapping microorganisms entering in our body. So these are physical barriers. The next barrier is physiological barriers. Acid in stomach, saliva in mouth and tears from eyes prevent growth of microorganisms. So they are the physiological barriers. The third type is cellular barriers. Polymorphonuclear leukocytes, monocytes and natural killer cells can phagocytize and destroy microorganisms. So the cells of our immune system, they are there to kill the microorganisms, so they are cellular barriers. Fourth one is cytokine barriers. Virus infected cells secrete proteins called interferons, which protect non-infected cells from further viral infection. So, such substances like interferons produced by the cells, they are called as cytokine barriers because they protect the non-infected cells from infection. Now let us see the next type of immunity, that is acquired immunity. It is pathogen specific. See, in innate immunity we said that it is non-specific. On the other hand, acquired immunity, it is pathogen specific. That means it will give the immunity or the response against a particular pathogen, and that is why it is called as pathogen specific. It is characterized by memory. Acquired immunity has a memory. It means when any microorganism enters in your body for the first time, your immune system identifies it and gives a primary response against it. Now, this primary response is of low intensity, and when the same organism enters the body again after few days or after some time, then that time the body gives secondary response, and this secondary response is highly intense. Now why is it so? Because this is because of the memory of that pathogen with the immune system. When it comes in the body for the first time, that time the immune system remembers, it has a memory of that pathogen, and that is why when it enters second time, the secondary response is highly intense. The cells involved in this acquired immunity are B lymphocytes and T lymphocytes. These are the two types of lymphocytes, B and T, and both are involved in this acquired immunity. There are no barriers. In innate immunity we have seen 4 types of barrier, but here the cells are involved, and the names of cells are B and T lymphocytes. B lymphocytes produce antibodies to fight against that particular pathogen which has entered the body, while the function of T lymphocytes is only to help B cells to produce antibodies. T lymphocytes on their own do not produce antibodies. They only help B cells to produce antibodies. Let us see the structure of an antibody molecule. When any pathogen enters in the body, our immune system produces an antibody molecule to fight or to kill that pathogen. Now, this antibody molecule is nothing but a protein. This molecule is made up of 4 peptide chains. In the diagram, you can see that 4 peptide chains are there, and the molecule, it looks like English alphabet Y. Out of 4 chains, you can see the 2 are long ones and the 2 are small ones. The 2 small chains, they are called as light chains, which are on the outer side. They are light chains because they are small in size. The longer chains, which are on the inner side and Y-shaped structure, they are called as heavy chains. So, in all, an antibody molecule has 4 peptide chains, 2 small, that is light, and 2 heavy. So, you can represent the antibody molecule as H2 L2. Now, in this structure, you can see there are two regions. This tip portion of the heavy chain and the light chain. This region is called as variable region. And in this variable region, the antigen molecule binds with this. It is mentioned there, that is, it is antigen binding sites are there on that. The disulfide bridge, you can see along this molecular structure, there are disulfide bridge present. Now, there are different types of antibodies. Antibody molecule is also called as immunoglobulin, and that is why it is abbreviated as Ig. So, the types of antibodies can be written as IgA, that is immunoglobulin A, immunoglobulin G, immunoglobulin M, and immunoglobulin E. Let us see the types of acquired immune response. Acquired immune responses are of two types. Humoral response. Antibodies produced by B lymphocytes are found in blood, and hence this is called as humoral immune response. Humor, the word meaning is fluid. Blood is a body fluid, and in this body fluid the antibodies are found, and these antibodies are produced by B lymphocytes, and hence such a immune response is called as humoral immune response. The second is cell-mediated immune response, or it is also called as cell-mediated immunity. T lymphocytes are responsible for this. Note the point that in humoral response, as we are saying, antibodies are there. So for antibody production, both types of lymphocytes, T and B cells, are important, while in cell-mediated immune response, only T lymphocytes are responsible. There is no any role of B lymphocytes in cell-mediated immune response. Now, where this cell-mediated immune response is important one. Before organ transplantation, tissue matching and blood group matching is done. Like we know that while in blood group matching, the blood groups are decided because of the antigen which is present on your RBCs. Same way, on your tissues, there are certain antigens, and the matching of these antigens is important before organ transplantation. Like in blood group, the particular type of blood group is matching, then only you can give it to the patient. Same way here, matching of these tissue antigens is important. Cell-mediated immune response is responsible for graft rejection. So, if the tissue antigens are not matching, say for example the liver transplantation is done and the antigens on the liver of the donor and the recipient are not matching, then that graft, that is the liver which is transplanted, will be rejected because the immune system gives the cell-mediated immune response, and these graft will not be accepted. Now, for the acceptance of this graft, doctors generally give immunosuppressant drugs. That means these medicines, they keep the immune system suppressed so that the cell-mediated immune response will not be given and the organ which is transplanted will remain in a proper way in the patient's body. Active and passive immunity. Active immunity is the one when the host body is exposed to a particular pathogen, maybe in the weakened form or dead form, or any protein of that pathogen is injected into the body of the host. That time the immune system of the host gives a response and produces antibodies against that particular pathogen. This is called as active immunity, as the body of the host is actively taking part in producing the antibodies against the pathogen. This is called as active immunity. This active immunity is slow. It takes time and to give its full effective response. On the other hand, passive immunity is the one in which ready-made antibodies are directly given to the patient. Here, the antibodies are not produced in the host or patient's body. They are prepared somewhere outside and ready-made antibodies are injected into the patient's body. So definitely this is quick and effective response. Now certain examples we will see. Colostrum, which is present in the mother's milk, contains antibodies, and these antibodies protect the infant. That is why it is said that breastfeeding is good for the infants, because it provides protection through the antibodies to the infant. Fetus also receives some antibodies from mother through placenta. When the fetus is growing in the womb of a mother, that time it receives some antibodies, and these antibodies protect the fetus.